Today I'll show you a complete workflow to get pure, viable, and functional astrocytes from adult mouse brain in half a day. Automated steps and standardized reagents are a prerequisite for reliable and reproducible results. With this step-by-step video tutorial, this challenging experiment will become easy and successful. There are 6 main steps. 1. Standardized tissue dissociation. 2. Debris removal. 3. Red blood cell removal. 4. Magnetic isolation of astrocytes. 5. Analysis by flow cytometry. 6. Cell culture. The protocol may be used for mouse brain tissue from P7 onward and any adult age. Let's get started with the tissue dissociation. Miltenyi Biotech's Adult Brain Dissociation Kit has been developed for the gentle, rapid, and efficient generation of single-cell suspensions from adult brain, considering the special needs of sensitive brain cells. The Adult Brain Dissociation Kit was developed for use with the GentleMax Octo-Dissociator with heaters. The automated process has been specifically developed for this purpose and is key for gentle dissociation. For animals younger than P7, we recommend using the Neural Tissue Dissociation Kit, P. To get started with this protocol in your lab, prepare 100 milliliters PBS and 50 milliliters PB buffer containing PBS with a pH of 7.2 and 0.5% BSA. You can achieve this by diluting max BSA stock solution 1:20 with PBS buffer. Keep the buffer cold. You will also need Dolbeco's phosphate-buffered saline with calcium and magnesium. When working with the Adult Brain Dissociation Kit, you first need to dissolve the lyophilized powder in the vial labeled enzyme A with 1 milliliter of buffer A. Do not vortex. This solution should then be aliquoted and stored at -20 degrees Celsius for later use. Avoid repeated freeze-thaw cycles. For astrocyte separation and cultivation, we recommend dissociating at least 800 milligrams of mouse brain tissue. Therefore, we double the volume of the enzyme mixes as we will dissociate 2 brains. Prepare enzyme mix 1 for 2 mouse brains by combining 100 microliters of enzyme P with 3,800 microliters of buffer Z. Prepare enzyme mix 2 for 2 mouse brains by combining 20 microliters of enzyme A with 40 microliters of buffer Y. A maximum of 500 milligrams of brain tissue can be processed in 1 C-tube. Therefore, we will need 2 C-tubes. Transfer 1,950 microliters of enzyme mix 1 into each GentleMax C-tube. Dissect the mouse brains and wash them in DPBS. Place the brains on a petri dish and cut them into 8 sagittal slices using a scalpel. Transfer the tissue pieces of 1 brain into 1 C-tube containing enzyme mix 1. Transfer 30 microliters of enzyme mix 2 into each C-tube. Tightly close the C-tubes until you feel and hear a click. Attach the 2 C-tubes upside down onto the sleeve of the GentleMax Octo-Dissociator with heaters. Make sure that the sample material is located in the upper right of the rotor blade. Run the GentleMax program 37C ABDK01. After termination of the program, detach the C-tubes. Optionally, you may centrifuge briefly to collect the sample at the bottom of the tube. To remove large cell aggregates from the sample, pass it through Max Smart strainers with a pore size of 70 micrometers. 1 Max Smart strainer can be used for 1 adult mouse brain, so you will need 2 of them. Wet the filters with DPBS buffer before use. Transfer the dissociated tissue from the C-tube to a Max Smart strainer placed on a 50 milliliter tube. Apply 10 milliliters of cold DPBS to each Max Smart strainer. Discard the Max Smart strainers and centrifuge the cell suspension at 300 times G for 10 minutes at 4 degrees Celsius. Aspirate the supernatant completely. Next, we will get rid of the cell debris that's still remaining in the cell suspension. For this step, it is mandatory to use a centrifuge with a swinging bucket rotor rather than a fixed angle rotor. This is important to obtain different phases. For 2 brains, we need 1,800 microliters of pre-cooled debris removal solution and a total of 10.2 milliliters of pre-cooled PBS. Resuspend the cell suspension derived from 1 mouse brain carefully with 3.1 milliliters of cold PBS. Transfer the suspension to a 15 milliliter tube. Do not vortex. Add 900 microliters per brain of cold debris removal solution and mix well. Overlay very gently with 4 milliliters of cold PBS. Pipette very slowly to ensure that the PBS phase overlays the cell suspension and phases are not mixed. Centrifuge at 4 degrees Celsius and 3000 times G for 10 minutes with full acceleration and full break. 3 phases will be visible after centrifugation. Aspirate the top 2 phases completely and discard them. Fill up with cold PBS to a final volume of 15 milliliters. Gently invert the tube 3 times. Do not vortex. Centrifuge at 4 degrees Celsius and 1000 times G for 10 minutes with full acceleration and full break. Aspirate the supernatant completely and discard. To eliminate erythrocytes from our target cells, we will use the red blood cell removal solution. First, dilute the cold 10 times red blood cell removal solution 1 to 10 with cold double distilled water. For example, dilute 100 microliters of red blood cell removal solution with 900 microliters of water. Store the prepared 1-time red blood cell removal solution at 2 to 8 degrees Celsius. Discard any unused solution at the end of the day. Resuspend the cell pellet from 2 mouse brains carefully in 1 milliliter of cold 1-time red blood cell removal solution. Do not vortex. Incubate for 10 minutes in the refrigerator at 2 to 8 degrees Celsius. Add 10 milliliters of cold PB buffer. Centrifuge at 4 degrees Celsius and 300 times G for 10 minutes. Aspirate the supernatant completely. Resuspend the cells carefully in 90 microliters of PB buffer by pipetting slowly up and down. Do not vortex. Determine the cell number either by using the absolute cell counting functionality of the Max Quant Analyzer or with a Neubauer chamber. In the next step, we are going to magnetically isolate the astrocytes with the anti-AXA2 microbead kit. The following volumes are appropriate for up to 1 time 10 to the 7th total cells, like in our experiment. When working with fewer cells, use the same volumes. When working with higher cell numbers, scale up the volumes accordingly. Add 10 microliters of FCR blocking reagent to the remaining 80 microliters of cell suspension. Mix well and incubate for 10 minutes in the refrigerator at 2 to 8 degrees Celsius. Add 10 microliters of anti-AXA2 microbeads. Mix well and incubate for 15 minutes in the refrigerator at 2 to 8 degrees Celsius. Wash the cells by adding 1 milliliter of PB buffer. Centrifuge at 300 times G for 5 minutes. Aspirate the supernatant completely. Place 2 ms columns in the magnetic field of the OctoMax separator. Prepare the columns by rinsing with 1 milliliter of PB buffer. The use of a precep filter is recommended. Suspend in 500 microliters of PB buffer. Apply the cell suspension onto the first column. Collect the unlabeled cells that pass through. Perform 3 washing steps with 0.5 milliliters of PB buffer each. Always wait until the column reservoir is empty before proceeding to the next step. Collect the total effluent. This is the AXA2 negative fraction. Remove the column from the separator and place it on a suitable tube. Pipette 1 milliliter of PB buffer onto the column. Immediately flush out the magnetically labeled cells by firmly pushing the plunger into the column. This is the fraction of enriched AXA2 positive target cells. To increase the purity of AXA2 positive cells, this fraction is applied onto a second MS column. Perform 3 washing steps with 0.5 milliliters of PB buffer each. Again, always wait until the column reservoir is empty before proceeding to the next step. Pipette 1 milliliter of PB buffer onto the column. Immediately flush out the magnetically labeled AXA2 positive cells into an appropriate tube. Analyze the cells by flow cytometry, for example with the Max Quant Analyzer 10. For a detailed protocol describing cell staining with fluorochrome conjugated anti-AXA2 antibodies, refer to the datasheet for the antibodies. Please note that anti-GLAST antibodies will not work in this case because of the initial papain-based dissociation step. For flow cytometry analysis, incubate the cells for 10 minutes at 4 degrees Celsius with the anti-AXA2 antibody. From 2 adult mice, you can obtain 8 × 10⁵ astrocytes on average, with a purity of 90 to 96%, depending on age and strain. To culture the isolated astrocytes, you need to prepare the appropriate medium, Max Neuro medium containing 2% Max Neuro Brew 21, 1% Penstrep, and 0.5 millimolar L-glutamine. Furthermore, you need a 24-well plate coated with polyl-lysine and laminine. This should be prepared in advance. Coat the plates by overnight incubation with 0.01% polyl-lysine at 37 degrees Celsius, followed by another overnight incubation with 10 microgram per milliliter laminin. Wash the plates 3 times with sterile H2O. Plate 1 × 10⁵ cells in 50 microliters of medium as a drop in the middle of a well. Let the cells settle down for 30 minutes at 37 degrees Celsius in an incubator. Add 450 microliters of medium. Maintain the culture by replacing half of the medium every other day. The cells will loosely attach during the first days of cultivation. Therefore, a cultivation period of approximately 1 week is recommended. After 7 days in culture, you may stain the cells for analysis by fluorescence microscopy. Congratulations, we're finished. I have shown you how to successfully isolate pure, viable astrocytes from adult mouse brain in half a day. The procedure can be used for total brain tissue or brain regions and yields cells of high purity and preserved vitality. Thanks for watching. To learn more about our neuroscience portfolio, visit miltonybiotech.com/neuroscience.